The final learning objective for this module is to describe signal transmission at a synapse. A synapse is the region where communication occurs between two neurons or between a neuron and an effector, which could be a muscle or a gland. So beginning with some terminology, and if a synapse is the region where communication between a neuron and a neuron or a neuron and an effector takes place, so this region here, the term presynaptic neuron, or simply presynaptic cell, refers to the neuron before the synapse. This is the neuron that carries the information or sends the action potential towards the synapse. The term postsynaptic neuron, which can also be a postsynaptic cell, is the neuron or cell that comes after the synapse. It's the cell that receives the action potential. Now, within the nervous system, there are two different kinds of synapses: electrical synapses and chemical synapses. At an electrical synapse, action potentials conduct directly between the plasma membranes of adjacent neurons through structures called gap junctions. So a gap junction is this structure here. This is our presynaptic cell. This is our postsynaptic cell. Each gap junction contains 100 or so tubular connections, which are these parts here, which act a little bit like tunnels to connect the cytosol of the two cells directly. So as ions flow from the cytosol of one cell through the connections to the other, it will change the membrane potential of that postsynaptic cell, and the action potential can continue. Now, gap junctions are common in visceral smooth muscle, in cardiac muscle, and in the brain. The benefits of an electrical synapse is that it allows for fast communication, as that action potential conducts directly through these gap junctions, whereas for chemical synapses, which we'll talk about next, there are more steps involved. Because of the fewer steps and the direct communication of these action potentials from one cell to the other, the electrical synapses also allow for communication to be more synchronized or coordinated. And this is important for things like the muscle of the heart or the gastrointestinal tract, where we need to have really precise, controlled, coordinated contraction to allow the blood to pump around the heart in the correct order, and for food to move easily down the digestive tract. At a chemical synapse, although the plasma membranes of a pre- and a postsynaptic neuron are close, they do not touch. So we have our presynaptic neuron here, we have our space, and we have our postsynaptic neuron or cell here. These two cells are separated by what we call a synaptic cleft, which is a microscopic space which is filled with interstitial fluid. Now, our nerve impulses can't conduct directly across the synaptic cleft, so a different form of communication must occur. So in response to an action potential, a presynaptic neuron will release a neurotransmitter, which is a chemical that diffuses through the interstitial fluid in that synaptic cleft and binds to a ligand-gated sodium channel, sometimes also just called a receptor, on the plasma membrane of the postsynaptic cell. The binding of this neurotransmitter to the ligand-gated sodium channel will open it, allowing the positively charged sodium ions into the cell and converting this chemical signal back to an electrical signal or an action potential. The time required for these processes to occur at a chemical synapse is about half a millisecond. And although that doesn't seem like a whole lot of time, it is considerably slower and therefore less coordinated than an electrical synapse. So looking at the steps involved in the transmission of an action potential at a chemical synapse, and this is something that you will need to know. And the first step is that the action potential moves down the presynaptic neuron. It ends up at the axon terminal, which, remember, is just the very end of the axon. As the action potential reaches the axon terminal, it causes a change in the membrane potential, which causes the opening of voltage-gated calcium channels. Calcium then moves down its concentration gradient and into the cell. The influx of calcium then signals to our synaptic vesicles, which are filled with neurotransmitter and found within the axon terminal. This causes the vesicles to move towards the plasma membrane of the presynaptic neuron, and these vesicles will merge with the plasma membrane and release the neurotransmitter into the synaptic cleft via exocytosis. Lastly, the neurotransmitter will diffuse across the synaptic cleft, bind to the ligand-gated sodium channels or the receptors on the plasma membrane of the postsynaptic neuron or cell. The binding of the neurotransmitter to the ligand-gated sodium channel will cause it to open. Sodium rushes into the cell. It changes the membrane potential, and the action potential may then propagate down into the next cell. Now, when a neurotransmitter binds to a ligand-gated sodium channel or just a receptor, it can have either an excitatory or an inhibitory effect. A neurotransmitter that causes depolarization of the postsynaptic membrane is excitatory because it brings the membrane closer to threshold. This is called an excitatory postsynaptic potential or an EPSP. A neurotransmitter that causes hyperpolarization of the postsynaptic membrane is inhibitory, as it takes the membrane further away from threshold. This causes an inhibitory postsynaptic potential, or an IPSP. Now, it's important to remember that a single neuron does not only receive input from a single synapse. A typical neuron will receive input from between 1,000 to 10,000 different synapses. So therefore, while a single excitatory postsynaptic potential is unlikely to initiate an action potential, it will make the postsynaptic membrane more excitable or bring it closer to threshold. And with summation of the excitatory or inhibitory input from all of the synapses, an action potential may then be generated. So summation of all of the input to a neuron is integrated within a region of the axon called the trigger zone, sometimes also called the axon hillock. And this part of the neuron will determine, based on the summation of all of the excitatory and all of the inhibitory input, whether or not threshold is reached, and therefore whether or not an action potential will be generated. Now, lastly, after the neurotransmitter has been released into the synaptic cleft and bound to the receptor or the ligand-gated sodium channel, that neurotransmitter needs to be removed so that the postsynaptic neuron or cell is not constantly being depolarized. In the nervous system, there are 3 ways in which that can be done. So, firstly, many neurotransmitters are actively transported back into the neuron that released them. This is known as reuptake, or transported into neighboring neuroglia. The neurotransmitter can then be recycled into new synaptic vesicles and can be released again. Secondly, some neurotransmitters are inactivated by enzymes. So an enzyme will change the shape of the neurotransmitter so that it can no longer bind to its specific ligand-gated ion channel. And then thirdly, some of the released neurotransmitter will simply just diffuse away from the synaptic cleft. We have to remember that all of this here is just interstitial fluid. And so not all of the neurotransmitter that is released will actually end up at that ligand-gated channel or on the receptor.